Welcome from GTA, Geospatial Technology Associates. In this video, we're going to do an introduction to spectroscopy for hyperspectral imaging. And in this picture, we can see an aircraft. This is a demonstration of where hyperspectral imaging is used. It's collecting hyperspectral imaging, processing it on board, using our Ninja software, and sending the output to a laptop, which could be placed anywhere in the world over HTML, with the results from processing that data. My name is Bill Basener. I'm the president of Geospatial Technology Associates. First, a little bit of definitions. I talk about digital images. Hyperspectral imaging is a collection of digital images. A digital image can be created using any measurable combination of bands or wavelengths of light. Visual digital images, which are what you're used to taking on a cell phone or a visual camera, things we're used to looking at, they collect images in three bands. There's a red band, which is around 650 nanometers for the wavelengths of the light, green band, which is wavelengths of light around 550 nanometers, and blue, which is around 450 nanometers. But there's no other reason that we can't collect imagery with bands at other wavelengths. And so in multispectral images, we collect a number of different wavelengths of light. Here's an image from the Worldview satellite, which has 16 bands. And the bands that we collect are usually in these visible from 450 to 650 nanometers on out into the infrared, which is the shortwave infrared and near infrared. And that goes out typically to about 2,500 nanometers. And a hyperspectral image, the way we define hyperspectral images, there's a couple of different ways people define them, but a good way to think about it is it's an image that has enough band to perform spectroscopy. And spectroscopy is the ability to distinguish different types of chemicals from each other. And usually this occurs when you have somewhere in the range of 50 to 400 bands. So we're going to talk about spectroscopy. And to talk about spectroscopy, spectroscopy is the measurement of the amount of light that's reflected or emitted from different materials. And so here is an example that we can associate with easily. 3 different types of fruit: an orange, an apple, and grapes. Light coming down on the fruit has all the different wavelengths, a full rainbow as we see it of light coming in. We see it as white when it has all the different wavelengths combined evenly. And the fruit will reflect different bands of light or different colors in different amounts. So an orange will reflect a lot of red, medium amount of green, and very little blue, where the apple will reflect a lot of red and not much green and blue, and the grapes similar to the apple. And so what we do is when we're doing spectroscopy, we measure the percent of light that's reflected off the material in percentages generally. And what we're going to be talking about is in percentages for reflected light. And so this is 80% of the red that comes into the orange gets reflected, 40% of the green is reflected, and 20% of the blue. And there are the numbers for the other types of fruit. What we generally do is we plot these percentages on a graph. And so here's the blue, the amount of green, the amount of red. And then this plot here we typically refer to as a spectrum, or these collection of percentages as a spectrum. When we talk about those collection of percentages for a specific material, we'll often call that the signature for that material, because that helps to identify that material from other materials. And so you see here gentle slope, and gentle slope is distinctive of the orange in contrast to the apple, which is horizontal and then a steeper slope. But the apple and the grapes would be difficult to tell apart in these wavelengths because their spectra in these 3 wavelengths is very similar. So these are what we would call confusers for each other. Now here are a couple of spectra for different fabrics, and the blue and green and red amounts, very similar to the plot we had before, are shown over here. And so you can see there's 2 red fabrics and 1 yellow fabric. And here's the yellow fabric. Here's visual pictures of the 2 red fabrics that these are spectra for. The red fabrics don't reflect much in the blue or the green, and they reflect a lot of red, whereas the yellow fabric reflects a lot of green and red, and that's why we see it as yellow. The interesting thing about these fabrics is you can see when we look out in the infrared regime here from about 700 out to about 2,500, the nylon fabrics have very similar dips and rises and peaks, and we refer to these dips and rises as peaks as features. They help us distinguish different materials. And so what's causing these dips and rises and peaks here is the light comes in incident upon the material at a certain wavelength. If that wavelength causes vibrations within the molecules, then that wavelength will be absorbed. If it doesn't cause vibrations, then it'll be reflected back. So these series of rises and peaks you can think of as reflections of the specific types of chemical bonds that are present within the materials you're looking at. So the two nylon fabrics, the yellow nylon and the red nylon, very similar chemical structures through those nylon polymers, and so the features are very similar through here, whereas the red cotton will have a different set of features or a different set of peaks and valleys. And here, just a little bit of terminology, spectrum is a measurement of light from the material, and then when we're going to use that spectrum as an identifier for that type of material, we call that a signature. Now here's another example of where somebody might use spectroscopy. And so this is an example. An envelope comes in the mail in this example, and it has some white powder in it. We want to determine if that white powder is nefarious or is dangerous. In this example, we're going to propose maybe that white powder is anthrax, and we want to match that white powder to other white powders it might be that are not nefarious. And so in this somewhat simplistic example, we think it might be anthrax, or here's a flour signature, it might be flour, or it might be powdered sugar. So these red and green and blue plots are specific spectra for the three different materials. And the anthrax, what we're using here is anthrax. It's actually a gypsum signature, and this isn't actually anthrax on the plate here. This is baking soda as a filler for this demonstration. And so what we would do is we would collect a spectrum off the white powder from the envelope, and then try to compare it to these three different known signatures to determine what type of material is present in the envelope. And if you haven't yet been able to compare the features here to those to determine what material is in our envelope, go to this slide. We have the same colors, powdered sugar shown in red, flour in green, our stand-in gypsum for anthrax shown in blue, and then the powder from the envelope is shown in black. And if you follow these features, the features for the black powder in the envelope match very well to the powdered sugar. And so we can say we've identified this white powder in the envelope as being powdered sugar and not flour and not anthrax or gypsum. So that's a good example of where we want to use spectroscopy. And when we come to hyperspectral images, we say we've collected hundreds, typically of bands, enough to do spectroscopy. So this is a hyperspectral image. You can download it from the website shown below. And just to look at a couple of different spectra from this image. So here is a spectrum from field grass. And you can see there is a peak here. This is at the green band, and that's why grass looks green, and then a drop. And then this rise here is very typical for chlorophyll. And then there's a series of features here that are typical for chlorophyll and the cellulose and materials that are present in vegetation. Next spectrum we have here is soil. This general shape here is typical for soil. These very sharp peaks have to do with noise in the image or atmospheric water bands that are causing this measurement. So those sharp peaks aren't true to the material. Those are because of the collection configuration for the image. This spectrum is a spectrum from evergreen trees, and you'll notice it's very similar to the spectrum for field grass. The features seem to be a little bit exaggerated. The slopes are a little steeper. These two rises are a little larger, more exaggerated than those, because there's more chlorophyll here. And you'll also know it peaks out at about 800 versus 4,000. The evergreen trees are a darker color green, or a darker spectrum overall in comparison to the field grass, but the shape is similar. And then here's one more spectrum from this image. This is a red latex painted roof. You could tell it's red by looking at the rise here and the high reflectance in the red compared to green and blue. This drop right here is typical for a polymer. So we'd look at this object just from the spectra and say we could be confident this is probably a red polymer of some type. But it happens to be a latex painted roof. So thank you very much for watching this video from Geospatial Technologies. Have a good day.